Earlier we went over animal cell, covering some of the basic organelles. I didn't want to go over all of it because it's too much to absorb at once. This video we will go over the same one again, but faster, and then cover the ones we did not go over. The two eukaryotic cells are plant and animal cells. Eukaryotic cells all have nucleus. The nucleus is mainly to store DNA. It's composed of nuclear membrane, the same material made of cell membrane, and there's also nuclear pores, which allows the RNA to go in and out freely. It contains the DNA, and it also contains the nucleolus, which makes ribosomes. Then there's the cell membrane, which is composed of a phospholipid bilayer similar to a city border. A zoom in of the cell membrane, we see a phospholipid bilayer. The inside is hydrophobic while the outside is hydrophilic. That means it likes water. The great thing about the cell membrane, and because the fact that it's hydrophobic inside, allows it to separate waters, allow it to create a separation between the inside and the outside, keeping all the organelles intact together. We take a closer look at one individual phospholipid. So we see that the inside, which is hydrophobic, is composed of fatty acids, while the outside is hydrophilic, composed of glycerol. Do remember that cell membrane, the phospholipid bilayer, contains proteins. Proteins have many functions on the cell membrane, such as transportation, receptors. Cell also has mitochondria, mainly in animal cell. There's more in animal cell, especially more in active cell. So if you exercise and you're an active person, then you would have more mitochondria. Mitochondria has a double membrane and it has its own DNA, which is a very unique quality which we'll learn later on. Then there's a Golgi apparatus. In a city, it's the same thing as a delivery structure, like a truck corporation. The purpose of the Golgi apparatus is to help transport materials, deliver the proteins, send out where it needs to go, create vacuoles for storage of either food, water, or waste. So the vacuole is like a storehouse. Then there's a lysosome, which is found only in animal cell. It's designed to break down stuff, like for example to destroy invaders such as bacteria and also destroy dead cell parts. We also have ribosome, which makes protein. Ribosomes are made from the nucleolus. Most of the ribosomes are attached to the rough ER. The endoplasmic reticulum ER is just stacks of membrane. There are two, rough and smooth. Rough ER has ribosome attached to it. Smooth ER mainly detoxify poison, so you have plenty of these in the liver. It also creates lipids from carbohydrates. All the empty spaces in the cell is a cytoplasm. Cyto is cell and plasm is substance. Students easily misunderstood, sometimes thinking that the cytoplasm is the filling. It's not. The liquid the organelles are expended in is actually called the cytosol, not the cytoplasm. The cytoskeleton provides support for the cell. You know, when you walk into those big factories and you look up and you see all those big metal beams out in the open that holds the whole factory? The cytoskeleton is like that. Cyto means cell, and remember that because you'll see it again a lot. So it's a cell skeleton. Remember cytokinesis, that process that occurs after mitosis when a cell split. Animal cell has flagellum for moving. Plant cells don't because they don't move. Animal cell also has lysosome, but plant cell doesn't. And we'll learn why in the future. Also in animal cell is centriole. They are responsible for splitting the cell during the process of mitosis. The centriole form a crew of two and are known as centrosome. In plants, it's called microtubule organizing center. This you have to be able to recognize for your test. We take a closer look at the flagellum. There are two parts to it. One is the flagellum strand, and the other is the basal body, where the flagellum is connected to the cell. Kind of like a rowboat, the paddle has two parts, the flat ends that extend in the water and the handles for rowing. The inside of the flagellum, the tip, is made of microtubules organized in pairs in a rule called 9 and 2. The 2 being the 2 in the middle. The basal body, the part that's connected to the cell, the microtubules are organized in a ring of triplets. Peroxisome are usually found near mitochondria and chloroplasts. It can produce hydrogen peroxide, which is dangerous, but it's used for breaking down toxic stuff. So you have lots of these in the livers too. If there's too much hydrogen peroxide, peroxisome can convert it to water. As a reminder, Mother Nature is so cool because she programmed everything to be so balanced. Remember how plants use stomata to control the exchange of gas at the same time it also creates negative pressure to pull up the water? It's a win-win situation. And now this, take peroxisome for an example. It can produce hydrogen peroxide to break down toxic stuff, but it has a safety switch, so if there's too much hydrogen peroxide, it can convert it into water, a safe neutral substance. Let me put into perspective. The nuclear plants that produce energy for a city uses radioactive nuclear power, but if anything goes wrong, boom. See how mankind is so unprepared? With modern nature, there's a safety switch for everything, and you'll see this as the more we learn.